Okay, hello everybody. Good afternoon. You can hear me and see the slides, right?
Yes.
Excellent. Beautiful day today. Okay, so 2 weeks from today is the day, October 30. We're going to have the second exam. And by now, I hope that you're all very familiar with Exam Soft. The exam 2 is not available yet. So I will let you know when you can download it onto your computer. But I just want to remind a few things. Because before you come to the auditorium on the 30th, make sure that your computer is fully charged and it's working properly. Remember, we are not technicians, right? The proctors, this is very important. The proctors and we're not technicians. We don't know how to solve software problems. So everything, this is your second exam, so you should know what you need, and hopefully everything will be working. And if it's not working, then you cannot take the exam, and you have to take a makeup at with the final. And you can only take one makeup. So this is very important that you remember so that you come to the exam with everything working properly. And how can you test? Remember that the mock exam is always available. So you can test your computer, the smart device that you're going to be using for the exam. So please do these steps. And if you have any problems, here's the chat support for ExamSoft. I've used them myself and they are really excellent. They go step by step with an individual and help you. If there's a major problem that they cannot help, then email me, please. You also can contact them by phone. So remember these very important guidelines for the exam so that you come prepared. And it should be easy now because you've taken the mock exam and you've taken the exam, the first exam. So you are pros now with ExamSoft. And if there are any questions, I can answer at the end, please. And if you don't like these cartoons that were made by ChatGPT under my direction, you have everything written here. So you can choose this or this. Okay, so let's go on to physiology, which is what we are here for. So what are we going to do today? Today, we're going to focus only on the skeletal muscle. In the last lecture, we talked about smooth muscle and cardio muscle, smooth muscle cells and cardiomyocytes. Now, this whole lecture is going to be dedicated only to the skeletal muscle. It's the type of muscle that is most abundant in our body. Okay, so remember that there are some properties of the skeletal muscle that are very similar to cardiac muscle. So they're striated. That's the most, one of the characteristic, one of the properties of skeletal and cardiomyocytes, skeletal muscle fibers and cardiomyocytes. They have this striated appearance. And today you will know what is this? What's the mystery? Why is it that these cells have this appearance of light and dark bands? Okay, so here what I'm showing is a bone with a tendon and with the muscle attached to the bone via the tendon. So a muscle, when we talk about the muscle, a muscle has many, is made up of many muscle cells, which we call muscle fibers. So a muscle fiber, which is represented here, this is one muscle cell, and here it's represented by each of these small circles. So the muscle fibers, the muscle cells in the muscle are arranged in bundles. Those bundles are known as fascicles. So you see here one group of muscle fibers, another group, and they are arranged in these bundles. They're kept in place by a layer of connective tissue known as perimysium. And the outside layer of the muscle that is known as epimysium. But also each of the muscle fibers, each of the muscle cells is enveloped by connective tissue also. And this connective tissue is important to maintain the blood vessels and so on that are going to nurture these muscle cells. So they keep all these structures and cells and bundles all together. So you see here the muscle fiber, the skeletal muscle fiber, and it has several nuclei. This is different. Cardiomyocytes and smooth muscle cells, they only have one nucleus. The skeletal muscle cells have many nuclei, and they result from the fusion of the precursor cells, which are myoblasts. So many myoblasts come together and they form one muscle cell, which we call a muscle fiber. So the skeletal muscles are striated, they have these dark and light bands. They have many nuclei, and now another property is that they are under voluntary control. Remember last lecture, we said that the smooth muscle and cardiomyocytes are under involuntary control. We don't have voluntary, we cannot control the smooth muscle and cardiomyocytes voluntarily. There's a special branch of the nervous system that's called the peripheral nervous system that takes care, that regulates the activity of the cardiomyocytes and smooth muscle. But the skeletal muscle is controlled by the central nervous system. So we have absolute control of the skeletal muscle cells. Sometimes there are reflexes, and this means that the action that controls the movement of specific muscles where there's a need for a quick response does not reach the central nervous system, does not reach the brain, but it goes into the spinal cord. So the spinal cord and the brain make up the CNS. Okay, so some of
Professor,
the,
just to be
yes,
clear, the
yes,
endoms, I don't know how to say the endomysium, is that like the
myos,
yeah, is that like the in this case kind of
endomysium.
like is this like the cytoplasm or where it's like
No, no, no. This is, so this epimysium, paramysium, and endomysium, they're connective tissue. So there's like a tissue that surrounds either the whole muscle, and that would be the epimysium. Remember that we call periosteum, endosteum, and epimysium. Remember? So
yeah.
here now we transport that into the muscles, and we just change the the last part, the mesium, which means muscle. So the epimysium is outside the muscle fiber, the muscle, right? So you have a muscle like the biceps. That muscle is made up of many, many muscle fibers. That's connective tissue that's known as the epimysium. And it's actually a continuation of the fascia, deep fascia. Okay, then a muscle, this is not a muscle cell, this is a muscle that contains many muscle cells. the muscle cells are arranged in bundles. So this is one muscle cell, right? And each of these circles is one of these. So each of these cells, muscle cells or muscle fibers, is surrounded by a layer of connective tissue that is the endomysium. It's outside the cytoplasm.
Should keep playing.
Okay, so it's outside the plasma membrane. It's a layer of connective tissue outside of the plasma membrane of the muscle fiber. So that's what the endomysium is. And then you have the perimysium that keeps these groups of muscle cells together to form bundles that are known as fascicles. Okay? So epimysium, perimysium, endomysium, they're connective tissue. They support the muscles, but they are not part of the muscle cells. Okay, is that clear?
So, what's the difference between perimysium and the fascicle?
And what?
And the fascicle?
Oh, you'll see the next slide.
Oh,
So the
okay.
epimysium is a continuation of the deep fascia. You're going to see soon. Okay. Okay. So when the tendons are the structures that attach the muscle to the bone, and they have to have some kind of elasticity. So, because the muscle contracts and relaxes, and the tendon has to respond to this movement. So some of the tendons are flat, and we call those aponeurosis. So you see here in the hand, so you see in the foot, these tendons are flat, and those are known as aponeurosis. Okay, so here you see the deep fascia. So here's the tendon, and then you have a layer of connective tissue that is in continuation of the tendon, and it also encircles the muscle. But then you have a deeper layer, which is the epimysium, and that the epimysium is in very close proximity of the muscle. It keeps in place also the blood vessels and so on. So if you go from the surface to the deeper end, you have the epimysium, which is a layer of connective tissue on the outside, covers the entire muscle. So you have to understand that a muscle is made up of many, many, many cells. And here you see the fascia surrounding groups of muscles. So this is the connective tissue. Okay, then, so you go from the epimysium, then you go on to the perimysium, which is the next layer of connective tissue. And this perimysium is what keeps these bundles of muscle fibers together. And it holds or keeps in place the larger nerves and blood vessels. So the muscles need a lot of blood because they're very active. They need a lot of nutrients and so on. And then you have a deeper layer of connective tissue, which is the endomysium. And the endomysium, so this is a muscle fiber, one muscle cell here. So the endomysium, this is one muscle cell. The endomysium surrounds each muscle cell. So this is a way of keeping the nerve endings and capillaries very close to each muscle fiber. So each muscle cell receives its own blood vessel and its own nerve, and it's kept close by this layer of connective tissue known as the endomysium. Okay, so here is a histological preparation of skeletal muscle. So this is one muscle fiber, one muscle cell, another one, and this white structure here, this is the endomysium. So when you look at a steak, when it's raw, you see a lot of white matter there that surrounds the steak and surrounds in its inside, that's all connective tissue. It has nothing to do with the muscle fibers. It's there to protect the muscles, the muscle fibers. And you see that this skeletal muscle fiber has many nuclei. One here, another one here, another one here. And remember, I mentioned this last time. Why is it that a skeletal muscle has so many nuclei compared to cardiomyocytes? They only have one nucleus, and the smooth muscle cell also has only one nucleus. Well, the skeletal muscle cells, they are very active. The proteins inside the skeletal muscle, they are subjected to a lot of wear and tear, and they need a lot of replacement. So those proteins need a lot of transcription translation. So you have to synthesize all these proteins to keep the muscle fiber healthy. So that's why you have so many nuclei, because the cells require a lot of protein synthesis. Okay, so now let's look at one muscle fiber. This is one cell, one muscle fiber. The nucleus is not shown here. The nuclei are not shown here. Okay, so the plasma membrane, so we're not talking about endomysium. We're now going inside a muscle fiber. So close to the endomysium, you have the plasma membrane, which in the skeletal muscle cell, it's known as the sarcolemma. So sarcolemma equals plasma membrane of a muscle fiber. Then inside, you have the cytoplasm, like in all other cells. The cytoplasm in the muscle fiber is known as sarcoplasm. Sarcoplasm equals cytoplasm in other cells. Okay, so far so good. Now, I want to talk about the sarcolemma. So the sarcolemma has some invaginations that go inside the muscle fiber. And these are known as the T tubules. And you will see why this is important. So keep in mind that the architecture of a skeletal muscle fiber is amazing, because remember, the cell has to contract and relax and produce movement. And it all depends on the molecular events that occur inside the cell. So each muscle fiber, which is very tiny, but there's a lot of activity going on in there. Okay, so you see these structures here, these tubes that are projections of the sarcolemma, and they go inside the muscle fiber. And I will explain why this is so important. These are the T tubules. Okay, so then another specialized sarcolemma is the cell membrane, right? What's the difference between sarcolemma and sarcoplasm? So the sarcolemma is the membrane. Sarcoplasm is the cytoplasm. It's inside the plasma membrane. Okay, so what else is different in these skeletal muscle cells? Here in blue, of course, in a regular cell, it doesn't have things in color. So here in blue is the endoplasmic reticulum, the smooth endoplasmic reticulum. And it does not contain ribosomes, remember? This endoplasmic reticulum is known in the muscle cell, is known as the sarcoplasmic. So you can see that every time you have this prefix, sarco, you're talking immediately, you're talking about muscle. Okay, so this blue structure, which is the endoplasmic reticulum or sarcoplasmic reticulum, in the muscle fiber, it has huge amounts of calcium ions. So calcium ions are critical or pivotal for muscle contraction and relaxation. So keep this in mind. Calcium is very important, and you will see why. At the molecular level, why is it that calcium, this ion, is so important for us to move our arms and our legs and all the muscles in our body? Now notice that this sarcoplasmic reticulum that has lots of calcium is very close to the T tubules. And why? Why is this proximity? So you have on each side of the T tubule, this is the T tubule, you have this enrichment in a sarcoplasmic reticulum. And the T tubule with the sarcoplasmic reticulum on each side is known as the triad. Okay? So triad means the T tubule and the sarcoplasmic reticulum that is close to on each side of the T tube. Okay, what else is special in these cells? And it's only present in cardiomyocytes and skeletal muscle. But now I'm only talking about skeletal muscle to make it things easier. Okay, so here you have what we call a myofibril. So we have now here two prefixes. Sarco is associated with muscle, and myo is associated with muscle. So what is the function of the T tubules? I will explain soon. Now we're just talking about the structure, and then we'll go deeper and discuss the function of this amazing architecture. Okay, so inside the skeletal muscle cell are these cylinders. These cylinders are known as myofibril. So myofiber is the whole cell. Myofibril relates to these cylinders. So you think of a cell with lots of cylinders inside, and these are known as the myofibrils. And the myofibrils, you can see that they are striated. You have the dark here represented in blue, and the light bands that don't have the blue. So they're different proteins in here. So a myofibril, one of these cylinders, is made up of different proteins that are known as myofilaments. So we have thin and thick filaments. So the thick and thin filaments are made up of different proteins. So you have this hierarchy of architecture where you have the myofilaments, which are proteins that are arranged in these cylinders. These cylinders are known as myofibrils, and the myofibrils are inside the myofiber, the cell. So each cell has many of these myofibrils, and they all respond in unison at the same time when there's a signal for the cell to contract. So all of these hundreds of myofibrils, these cylinders, when there's a signal that comes from a nerve cell known as motor neuron, when the signal arrives and activates one of these muscle fibers, all of these cylinders will respond in unison at the same time. Okay, so here, of course, you don't see the lysosomes, you don't see the nuclei. This is just the structures that are important for muscle contraction. I left out many of the organelles. We just concentrating here on the T tubules, which are an invagination of the sarcolemma. We're talking about the sarcoplasmic reticulum that has high concentrations of calcium. And we're talking about these cylinders that are the myofibrils that contain the thick and thin filament. Okay, so here there's another view of these T tubules. So you see, these are not pores. These are this is the sarcolemma, right? And these are the openings to these T tubules. So this is still sarcolemma. This light yellow or beige structure, this is a T tubule, and it's part of the sarcolemma. So that means if this is a hole and this is a hollow tube, that means that the extracellular fluid goes inside this T tubule. And you see that on each side of the T tubule, here represented in yellow, this is the sarcoplasmic reticulum that is highly enriched in calcium. And remember, why is this structure so important? So it's important because it allows ions that are in the extracellular fluid to come into the T tubule and to signal the sarcoplasmic reticulum to release calcium. So that's the function of the T tubule. So the T tubule has extracellular fluid that has ions that can go through the inside of the T tubule, which is hollow, and they send signals to the sarcoplasmic reticulum so that calcium is released. And then calcium floods the cell and induces muscle contraction. So here it might seem very complicated, but I'm going to go step by step on this. So this is just a structural slide showing the sarcolemma, which is the plasma membrane, the T tubule, which is an extension of the plasma membrane that goes inside the cell, and it's hollow and allows the extracellular fluid to go through this hollow tube inside. And then there's receptors on the membrane of the T tubules that communicate with the endoplasmic reticulum, which is the sarcoplasmic reticulum, to signal to induce the release of calcium. Okay, so this slide just shows you the hierarchy of these different structures. So let's go from the smallest to the largest. So the smallest are the myofilaments. These are the proteins that make up the thick and thin filaments. So the thick and thin filaments are components of a myofibril. A myofibril are these cylinders right here that are enveloped in this endoplasmic reticulum. So here you see this thick and thin filaments, and this is a myofibril. And then you go up, so a larger structure, which is the muscle cell, the myofibril. Okay, so let's go a little bit into more detail. So here you see the endomysium. Remember, this is the connective tissue that surrounds the muscle fiber. Then you have the sarcolemma, which is the plasma membrane that is in very close proximity to the endomysium. Now we go inside the cell. We see the sarcoplasmic reticulum, and you see the T tubules, which are extensions of the sarcolemma. Now let's look at the myofibril. So the myofibril, one of these cylinders, right? You have here 1, 2, 3, 4, 5. These cylinders are located inside the muscle cell. This is what they look like. This is a myofibril. So they're proteins that are bundled together and that make up this cylinder. Now we go zoom in even more. Now we look at the myofilaments. And now here you see why you get this appearance of dark and light bands. It's caused by the proteins that make up the myofilaments, the thick and thin filaments. So the blue, which, in a real cell doesn't have a color, but this is the thick filament. It's made up of a protein known as myosin. This is the thin filament, sorry. The blue is the thin filament. It's made up of actin. And then this one here, this is the thick filament, and this is the one made up of myosin. So you have these thick and thin filaments, you see here, that give this appearance of striation. So this striation that is visible or apparent in the myofibril in one of these cylinders is caused by the components, the proteins that make up the myofilaments. Okay, so let's go into more detail. So this is an electron micrograph of one of these structures. So within a myofilament, within a myofibril, you have these units that are repeated many, many times, and that they are limited by these other proteins that separate one unit from the other. This unit is known as a sarcomere. So within a myofibril, you have thousands and thousands of these sarcomeres. The sarcomere is a unit of the myofibril. You see here's the electron micrograph. So no color, right? So you see the dark area contains the thick filament and the light area contains the thin filament. So here there's no color, but you can definitely see the striation, the thick and thin filament. So this is a unit that makes up the myofibril. So you have these units repeated all along the filament. Do the sarcomeres make up or made up of myofilaments? Yes, correct. 100% correct. Okay, so again, let's zoom in. But before that, I want to show here. Here you see a motor neuron represented in yellow. So this motor neuron comes all the way from the spinal cord. It has the cell body in the spinal cord, at the spinal cord, and it comes all the way down. The axon divides into these several axon terminal, which are going to modulate the muscle fiber, the muscle cell. This is a muscle cell. And the connection between a motor neuron and the muscle fiber is mediated by calcium ions. So you see here already two functions of calcium. Here, where you have lots of calcium here that is going to be released when there's a signal carried through the T-tubule, and that's essential for muscle contraction. But also you see here calcium, very important for the release, for the connection between the motor neuron and the muscle fiber. Okay, so now let's zoom in a sarcomere. So remember that the thick filaments make up the dark area of the sarcomere and the thin filaments make the light area. So here you see represented this thick band that is the thick filament and it's made up of myosin. And then here you see the thin filaments that give the appearance of the light zone. And these are made up of three proteins, but one is actin. Okay, so a sarcomere is limited by this Z line. This Z line is made up of many proteins, one of them being actin. So this Z line maintains the sarcomere in place. Limits is the limitation of the sarcomere. So you have two Z lines, and their function is to where the thick filament and the thin filament, they are attached to the Z line. So this is the unit, the molecular unit that is responsible for our muscles to be able to contract and relax and to respond to stimulus by the motor neuron. So this is a very, very important unit, and we're going to see how does it work. Okay, so thick and thin filaments. So a sarcomere has different areas, and we like to label them because they have different functions. Okay, so let's say, what is the A band? The A band is the area within a sarcomere that contains the entire thick filament and some of the thin filament that overlaps with the thick filament. So that's the A band, it goes from here to here. So it contains the entire thick filament and some of the thin filament that's overlapping. What is the I band? The I band is the area that contains the Z line and the thin filament. Okay, now the M line, this is made up of proteins and it stabilizes the thick filament. So it's right in the middle of the thick filaments. Now here is a very, very important area that you need to remember. That's the H zone. The H zone, you see, this is this space here that now it's showing that it only has thick filament. But the H zone can change in width. And you will see when the muscles contract, these proteins never, ever, ever change in size. The size is always the same. So you might ask, but how is that possible? Is the Z line the same as the Z disc? Yes, the same, synonymous. So how is it possible that the muscle contracts and relaxes and these proteins never change in size? No, because what they do, they slide upon each other. So that means that this H zone can change in width. So the H zone is the area you see here that contains only the thick filament. So this area, if there's more overlap of the thin filament over the thick filament, the H zone is narrow. Now, if the thin filament spreads out, then the H zone is going to be wider. But remember, always keep in mind that the size of the thick and thin filament never changed. The length of the thick and thin filament never changes. What changes is the overlapping of the two structures, the thick and the thin filament. Okay, so here again, you see the sarcomere. Here in red are the Z lines. Here's the M line, like here. And this is the H zone. So the I band only has thin filament, right? And the H band, the H zone, H band, H zone, is this area that only has thick filament. Okay, now here you see a muscle fiber, and you see this is the motor neuron, and they're not touching. The motor neuron does not touch the muscle fiber. There's a little space known as the synaptic cleft. And you will see how is it possible that the motor neuron sends a message to the muscle fiber if they're not touching each other. So the release of neurotransmitters from the motor neuron carry the signal to the muscle fiber. So each myofibril is made up of thousands of sarcomeres. And when there's a signal from the motor neuron, they all respond in unison at the same time. It's what we call an all or none response. So either all of them respond or none of them respond. So the all or none response refers to all the sarcomeres responding to the signal coming from the motor neuron at the same time. Yeah, so the M line is right here. So the M line is made up of proteins that stabilize the thick filament. You see here? Okay, so this is just another way of looking. So you see here, this is the electron micrograph. So this is the real thing. There's no color, but you can see that there's the dark area, which is the thick filament, and the light area, which is the thin filament. So no, you're going to see they both move upon each other. You're going to see in a little bit. You will see that. Yes, they can overlap, but their length doesn't change. Exactly. Okay, so now let's look closer to the composition of the thick and thin filaments. So here in blue, this is the thick filament. It's made up of myosin. And this is the thin filament made up of actin, troponin, and tropomyosin. So what are the functions of these proteins? Let's now concentrate on myosin. So myosin is the component of the thick filament. And it's made up of two peptides that intertwine, and it looks like a golf a golf stick, right? So they have this tail. So there's two polypeptides here represented in light lavender and dark lavender. And at the end, there is this structure that is the head. So the myosin protein is made up of these two polypeptides that have a tail and a head. And this head is very important. So the myosin, you can say it's an enzyme. And the activity of the enzyme is at the head. And this head has two activities at the same time. It's an ATPase. It binds ATP and it hydrolyzes ATP to release energy. Okay? So this is an enzyme. Myosin is an enzyme. And the head is where the enzymatic activity is located. So it's an ATPase because it hydrolyzes ATP to release energy. But also it has another function. It's where the actin, the fin filament, binds to, or the head of the myosin binds to actin. So the head of the myosin is an ATPase, but also has binding sites for actin. This is very important. So when the head is bound to actin, the fin filament, they form what is called cross bridges. So you see here many molecules of myosin. It looks like this rope structure with the head sticking out. It's very important that the heads stick out because they're going to form cross bridges with the fin filament, with the actin. Okay, let's look at actin. So actin is a smaller protein. It's globular, roundish in shape, but it polymerizes. So you get many of these units. Each of them we call a G actin. So each of these spheres represents one actin molecule, but they can bind to each other and form F actin, which is filamentous actin. So the fin filament has the actin, but also has these two other proteins known as tropomyosin and troponin. So you see, I mean, just think about this. In your cell, all this is happening. I'm moving my hands, and all this is happening in the muscle fibers, in the muscles of my hand. Okay, so tropomyosin, this rod-shaped protein, is covering the actin binding site to myosin. So when there's no signal, when there's no activation, the actin and the myosin are separated. And there's no binding of the heads to actin because tropomyosin is covering the binding sites for myosin. And then on top of the tropomyosin, you have troponin. Troponin has three peptides make up this protein. And its function is to bind calcium. Now you understand why you need so much calcium that is stored in the sarcoplasmic reticulum. So when calcium is released, it binds to troponin. Troponin changes the shape of tropomyosin so that it moves out of the way, so that the myosin binding site is exposed, and now the myosin heads can bind to actin. Okay, so these three proteins are architecturally involved. The design is such that one, the troponin binds to calcium. Once it binds to calcium, it causes a change in the tropomyosin. So it moves out of the way so that now myosin can bind to actin. And all this starts with ATP, with the hydrolysis of ATP and calcium. Okay. So I talked already about the actin, and you see the troponin, tropomyosin, and the troponin. Okay, so here you have a view, a side view of the actin, the fin filament, with the tropomyosin. And here you have the myosin with the heads. sticking out towards the fin filament. Okay, now just to give a few more proteins that make up this sarcomere, this unit. So you see here the M line, right? That keeps the thick filament, the myosin, in place. But now the myosin cannot just be floating around. It has to be stabilized. So it's binding to the Z line or Z disc. So the Z disc is made up of many proteins, one of them being actin. It has many others. And you see this protein here that has this shape. This is known as titin. It's one of the most abundant proteins, and it's very large in size. So what it does is stabilizes the thick filament and attaches to the Z line. Now you have nebulin. What nebulin does, it stabilizes the fin filament. So it attaches the fin filament to the Z line. So you have these two proteins that are structural proteins. They actually don't play a role in the sliding of the thick and fin filament. So titin and nebulin. And there's another protein. So there's many proteins here. I'm just calling out a few. That is known as dystrophin. So this protein, dystrophin, its function is to connect or attach actin to the sarcolemma, to the plasma membrane. So it stabilizes the position of actin in relation to the sarcolemma. And why am I pointing out dystrophin? Because there's mutations in this protein that lead to muscular dystrophy. And this is a condition that occurs mostly in boys. And this leads to the degeneration of muscles. And the children end up in wheelchairs just because of this mutation in this protein, dystrophin. That's why I want to emphasize it. Okay. So now you understand that when we're moving and doing all kinds of exercises, all these proteins that make up the sarcomere, that unit of muscle contraction, they're going to be subjected to a lot of wear and tear. So that feeling of pain after doing a lot of exercises is caused, one of the factors is the distortion and the degeneration of these proteins. So you have a torn sarcolemma, so that's the plasma membrane. You have damaged myofibrils. Remember, the myofibrils are the cylinders, and the disruption of Z disc. So a lot of damage can occur when you're doing exercise. It's not only the disruption of the sarcolemma, it also has to do with too much lactic acid accumulating in the muscle, but we'll go through this later. Okay, so one of the factors that contributes to the muscle sore or pain is the wear and tear of the sarcomere. Let's go at the microscopic or molecular layer. Okay, so we're done with the muscle contraction. So what happens now when a motor neuron sends a signal to the muscle fibers? So what you see here, this is a cross section of the spinal cord. So the spinal cord goes through the spine. And you see here two motor neurons, one in blue, one in red. And the little sphere is the cell body. So these motor neurons can be very long. Just think about the sciatic nerve. It goes all the way from the spinal cord, all the way down the leg. So, if you impinge it or something, it can cause a lot of pain. That's sciatica. So some of the axons can be very long, others not so long. But now let's look at the blue motor neuron. So you see that the motor neuron can divide into different axon terminals, into different divides into different lines, And each of them is going to control one muscle fiber. So you see here that the blue motor neuron is controlling 4 muscle fibers. 1, 2, 3, 4. Each of these is one muscle cell. Now, the red motor neuron controls also 4 muscle fibers. So you see that each motor neuron can control a group of muscle cells. So that is called the motor unit. The motor unit is defined by the motor neuron and the number of muscle fibers it innervates or it controls. Okay? So the blue motor neuron, so you have a motor unit made up of the blue motor neuron and the 4 muscle fibers that it controls, that it innervates. And the signal that goes along these motor neurons is an electrical impulse that is generated by ions that cause an action potential. Not the calcium doesn't cause muscle damage. The calcium plays a very important role in muscle contraction. But what causes the damage is you have all these proteins moving up and down very quickly, right? Over each other, overlapping, and it's that movement that causes the damage. Oh. Is it one neuron for one motor unit? Yes. One motor neuron and the muscle fibers that it innervates. Yes. One motor neuron, one or more muscle fibers. Okay. So here you have the motor neuron with the cell body and the axon. You see that the axon is in blue here. And you see that it has this yellowish cover. That is the myelin sheaths. Remember in multiple sclerosis, it's an autoimmune disease where the myelin sheaths are damaged by the white blood cells. So you can see that the myelin sheath is very important for the propagation of the signal along the axon. Okay, so once the motor neuron reaches the muscle fiber, you see that it divides into different axon terminals, but it's not physically touching the muscle fiber. So here you see the motor neuron again with the axon, and you envision an action potential, an electrical current coming down the axon and reaching the axon terminal, right? Reaching the axon terminal. What happens here? At the axon terminal, you have many vesicles here represented by these little spheres that contain a neurotransmitter, a chemical inside. In the motor neuron, the chemical that's inside these vesicles is always a chemical known as acetylcholine. Acetylcholine. Okay, so here's a motor neuron, and here's the muscle fiber. And you see that there's a space. So here you have a synapse. What's a synapse? A synapse is where a motor neuron is in proximity to the muscle fiber that it's going to modulate. And this is known as a neuromuscular junction. It makes sense. Junction because you have the two close to each other. Neuro because you have the neuron end, and muscular because you have the muscle fiber. So you see here that there's a space. They're not touching each other. There's a space that's known as a synaptic cleft. And this synaptic cleft is very important because here we're going to talk about calcium again. So when the action potential is traveling down, right? It's traveling down the axon. It reaches the synaptic terminal. It reaches the synaptic terminal. What happens here? When the action potential reaches the synaptic terminal, there are calcium channels here that open up in response to the action potential that is reaching here. When these calcium channels open, calcium gets into the synaptic terminal. And that's the signal for these vesicles, known as synaptic vesicles, to move to the edge of the plasma membrane. Once they go and move to the edge, they open up and release the neurotransmitter, the acetylcholine, by exocytosis. Remember, we talked about exocytosis. So here the events, the cascade of events is the action potential, reaching the axon terminal, opening up or triggering the opening of the calcium channels. Calcium floods the synaptic terminal, induces the movement of the synaptic vesicles towards the edge of the plasma membrane. The synaptic vesicle fuses with the plasma membrane and releases acetylcholine, the neurotransmitter, by exocytosis. Okay, so now you have the synaptic cleft, this space, filled with acetylcholine. What happens? So acetylcholine is going to diffuse throughout the synaptic cleft, so it's going to move around. And some of it is going to reach the sarcolemma of the muscle fiber, where there are receptors for acetylcholine. So receptors are proteins that are located at the plasma membrane or sarcolemma of the muscle fiber, and they are specific for acetylcholine. Okay, so here you see this another view of this. So you see here the motor neuron, you see the muscle fiber with the myofibers, and now you see here, zoom in, where you see the axon terminal, and when the acetylcholine, when calcium reaches, so this blue represents the axon terminal of the motor neuron. So you see the acetylcholine in the synaptic vesicles. So here, there's no signal. So they are separated and there's no acetylcholine. There's only acetylcholine receptors on the sarcolemma of the muscle fiber. So when there's a signal and then acetylcholine is released into the synaptic cleft, it diffuses across the synaptic cleft and it binds to acetylcholine receptors. And you see here sodium. Why is sodium here? Because the acetylcholine receptors are sodium channels. So what happens when acetylcholine binds to its receptor on the sarcolemma, it opens up sodium channels. And now it's the sodium channels that go in. That's the nicotinic bind to the, so nicotinic binds to the acetylcholine receptor. Actually, this receptor for acetylcholine is known as the nicotinic receptor. Okay, so you see all these events that happen all the time inside the muscle cells at the microscopic level. So we're talking about the molecular layer, the molecular level. Okay, now another term here, motor end plate. What is this? The motor end plate is the area where, of the sarcolemma, where it's in close proximity to the motor neuron. So motor end plate. It's this area here of the sarcolemma that is in close proximity to the motor neuron. Now you see here that the sarcolemma that is at the motor end plate has these folds. And these folds, they're there because they are filled, they have a high density of acetylcholine receptors. And why? Why do you need so many acetylcholine receptors? To generate a very strong electrical signal. Okay, so let's go to the next slide. Well, this shows the same. You see the motor neuron, the axon terminals. You see the myofibrils. We haven't discussed what's going on inside. But here, what I want to point out is the motor end plate. You see, here's the axon terminal with the synaptic vesicles filled with acetylcholine. Here you have the motor end plate, which is this area of the sarcolemma that is close proximity of the axon terminal. And you see that there are all these folds of the sarcolemma at the motor end plate, and they are highly, highly, highly enriched in acetylcholine receptor. So remember, motor end plate is the area at the sarcolemma of the muscle fiber that is in close proximity to the axon terminal. Okay, so now what happens here? What happens when acetylcholine is released and binds to the sodium channels, to the acetylcholine receptor, which is a sodium channel? Okay, now remember, when we go back to discussing the plasma membrane, we have a concentration gradient where on the outside you have lots of sodium ions, the inside you have lots of potassium. But in terms of electrical distribution, the outside is positive in comparison to the inside. And this only happens close to the plasma membrane. So the motor end plate is this area of the sarcolemma that is close proximity to the axon terminal. Okay, so here you have two things to remember. First, there's a difference in ion distribution across the plasma, on the both sides of the plasma membrane. Lots of sodium, lots of potassium. But inside, you also have proteins and phosphate groups that give it a negative charge. So the voltage across the plasma membrane, outside is positive, inside is negative, and in the muscle fiber, the potential across the plasma membrane is minus 90 millivolts. That is the RMP, the resting membrane potential. So the difference here, the voltage across the plasma membrane in the resting stage is minus 90 millivolts. Okay.
Professor,
Yes,
the notes say minus 60 millivolts.
we're going there. We're talking about the resting membrane potential. We're going to go to minus 60, but not yet. Okay? We are at the resting membrane potential, which is minus 90 millivolts. The minus 60, when it goes up to minus 60, that means that there's a signal and there's sodium coming in. So that's a signal of activation. We'll get there. Okay, so the minus 90 millivolts at the motor end plate is caused by the positive charge due to lots of sodium, a negative charge inside because you have more potassium inside, but that doesn't give you the negativity. It's mostly proteins which are negatively charged. So the voltage across the plasma membrane is minus 90 millivolts at the motor end plate. Okay, so here is just repeating what I just said. Remember the T tubules here. Here's where the sarcoplasmic reticulum, the sarcolemma, the tubule is an extension, a hollow tube that's an extension of the sarcolemma. Here you see the calcium ions coming in, here are mitochondria. You see the synaptic vesicles. And here again, you see the motor end plate. It has all these folds that are highly enriched in acetylcholine receptors. Okay, so now what happens? The acetylcholine receptor that is represented here by this purple structure, it's made up of different proteins that come together and form a sodium channel. Actually, it's a sodium and potassium channel. It allows sodium to come in and some potassium to go out. But the most important event is the movement of sodium from the outside, from the synaptic cleft. So this is the synaptic cleft, right? This is the axon terminal. And here's acetylcholine that's being released. and it binds to its receptor. Once it binds to its receptor, the channel opens up and sodium floods in, goes in. So what happens now? Here you have the minus 60 millivolts that you were talking about. Okay, so when there's no signal, this is the channel, which is also, it's a ligand-gated ion channel. So that means that the acetylcholine receptor here, it's also a nicotinic receptor because it binds nicotine. But let's not get there. The acetylcholine receptor at the motor end plate is a ligand-gated ion channel. What does that mean? That means that it opens in response to a ligand, to a chemical, and the chemical is acetylcholine. So that means that this protein is highly specific. It has a specific binding site for acetylcholine. So you need two molecules of acetylcholine to bind to its receptor, to this sodium potassium channel, but we were going to call it just a sodium channel for simplicity. When two molecules of acetylcholine bind, the channel opens and you see sodium rushing in, some potassium going out, but let's just concentrate on the sodium. Okay, so the resting membrane potential was minus 90 millivolts. Now you have sodium going in, so you're going to increase the voltage. So it's going to go up to minus 60 millivolts. And this is the end plate potential. So here you had the resting membrane potential. Well, where was it? Here, the RMP, that's represented here. That would be the resting membrane potential. So across the sarcolemma would be minus 90 millivolts. But here, once the sodium comes in, it goes up to minus 60 millivolts. So that's the end plate potential, or EPP. Okay? So you go from minus 90, which is the resting membrane potential, when the sodium comes in, because acetylcholine bound to its receptor, open up the channel, sodium goes in, and now it becomes less negative, the difference. So it goes to minus 60 millivolts, EPP. Okay, the page. Okay, this, I'm now focusing on this. Please bring up the page numbers at the end, okay? Okay, so now what you see here, you see the axon terminal with the synaptic vesicles. You see the motor end plate, right? This area of the sarcolemma that's close to the axon terminal. You see that it has all these invaginations, all these folds. And you see here, each of these little structures represents an acetylcholine receptor or the sodium channel. So you see that it's highly, highly enriched at the motor end plate. Okay, so what happens when there's a signal coming from the axon of the motor end plate? So acetylcholine is a neurotransmitter. The ion channel, the sodium channel, is an acetylcholine receptor. It opens up when acetylcholine binds to it. Sodium enters, and it changes the voltage from minus 90 millivolts to minus 60 millivolts. Okay? So this is all, this is the end plate potential, is the minus 60 millivolts. Okay, so here you see, now I'm going to make things a little bit more complicated. So I said that the acetylcholine receptor is an ion channel that is chemically regulated. So why do we say these are gated ion channels? Because they are closed, there's a gate, they are closed when they are not, when there's no signal. So the acetylcholine receptor is a ligand regulated ion channel. So here you see when there's no acetylcholine bound to it, the channel is closed. When acetylcholine binds to it, then it opens up. So it's a gated ion channel that depends on the binding of a chemical, in this case, acetylcholine. So it's a chemically regulated ion channel. But this type of ion channel is only present at the motor end plate. So what happens in the rest of the sarcolemma that is not close to the axon terminal? So that area of the sarcolemma has another type of sodium channel that is known as the voltage regulated ion channel. So what happens is when there's minus 90 millivolts, the channel is closed. You see, this channel has two gates. One is the activation gate, the other one is the inactivation gate. So now concentrate on the minutiae of this. Yes, acetylcholine binds to the nicotine receptor. So now let's look at this other channel. How does it open and close? So it's closed, the activation gate is closed, and the inactivation gate is open. When there's the resting membrane potential, which is minus 90 millivolts. Now, when there's the signal and sodium rushes in, the voltage is going to change from minus 90 to minus 60 millivolts. What happens here is that this voltage regulated, so it opens in response to a change in voltage, so going from minus 90 to minus 60. This gate opens and it allows sodium to go in. So now it becomes positive, right? This side of the sarcolemma becomes positive because you have a rush of sodium. So now it goes from minus 60 to plus 30. When it reaches plus 30, the inactivation gate closes. You see? So during this time, the inactivation gate is open. When it reaches plus 30 millivolts, the potential across the plasma membrane, the inactivation gate closes. Why does it close? So that there's no more sodium going in. So you're going from a resting membrane potential, which is minus 90, to the EPP, the end plate potential, which is minus 60 millivolts, that is going to be propagated to the sarcolemma that has no acetylcholine receptors, but it has this voltage regulated ion channel. So now sodium goes in. When the potential reaches plus 30 millivolts, the inactivation gate closes and there's no more sodium coming in. So here you see the same. How is it that this sodium channel responds to these changes in voltage? So you have a helix, an alpha helix present in this sodium channel that is sensitive to the changes in potential. So you go to depolarization. Depolarization is when it goes from minus 60 to plus 30. That's known, the phase, the stage is known as depolarization. And then you have the inactivation stage when the inactivation gate closes. So how does it know the change in voltage? Because it's like, you know, if you put electrodes, you can measure the electrical current. So there's an electrical current that is being propagated along the sarcolemma. And when it reaches this channel, the structure of the protein that makes up this channel, the alpha helix, is going to change when there's a change in voltage. Okay, so it's the folding of the protein, the alpha helix. Remember, that's the second level of structure that senses the change in voltage. Okay, so now you have here the two different channels, right? That they all concentrate on sodium, right? Here is a ligand binding ion channel, so it opens when a chemical, in this case acetylcholine, binds to it. It opens and allows sodium to go in. This channel is at the end plate, right? Motor end plate. Now here, so then the voltage, when it goes from minus 90, which is the resting membrane potential, to minus 60, then it's propagated, it goes along, the electrical current goes along, reaches the sodium channel, the voltage gated sodium channel, and now sodium goes in and reaches 30 millivolts. Okay, so what happens now? What happens? How can you reestablish now? How can you go back to minus 90, right? You're now at plus 30. How do you go now back to minus 90? Now it's when the potassium channel goes into place. So you have a potassium channel that is also a voltage gated ion channel. When a certain voltage, plus 30 millivolts, is at the sarcolemma, the potassium channel opens and it brings the potential back to minus 90.
Professor,
Yes?
I have a question. So I was just wondering, how does increasing the potential between the membranes lead to muscle contraction?
Yes. We are going to discuss that in the next lecture.
Okay.
Okay? How does this electrical signal now is converted to the sliding of the thick and thin filaments? That's what you're asking, right? We'll do that in the next lecture. Okay? So now here you see, I want to go to this slide here. Okay, so let's see the different stages. So here you have the arrival of the nerve signal. So here there's the resting membrane potential. It's still there on the plasma, on the sarcolemma, minus 90 millivolts. Then you have acetylcholine being released, binding to its receptor. So now you go from minus 90 to minus 60 at the motor end plate. Now, this minus 60 change in voltage is going to be propagated. Oh, so ligand gated. So you have the sodium going in. And now you have the sodium channel, voltage gated ion channel opening up until it reaches 30 plus 30 millivolts. And then that triggers the opening of the potassium channel, leading to repolarization. So the sodium channel causes depolarization, going from minus 60 to plus 30, and the potassium channel leads to repolarization. So this is so you see here now, if you would make a graph representing the change in voltage, so you reach minus 60, then it goes up to 30 plus 30. So this is when the sodium voltage gated ion channels are open, and then you get repolarization and the potassium channels open, and it goes back to minus 90 millivolts. And this propagates along the sarcolemma of the motor end plate. So this is an amazing phenomenon, a cascade of events that allows the communication between the motor neuron and the muscle fiber. Now, for the next class, we're going to see what happens inside the muscle fiber once this change in charge occurs. So I highly recommend that you see this video, YouTube video that explains very slowly and in detail the different steps that I just explained. Okay, so we're done. And see you Tuesday. Any questions? What would happen if the potassium channel would not release potassium? That means that the cells could no longer be activated, right? So that's called the refractory period. So there's a certain period where the cells cannot be activated No longer, because there's no differential distribution. Now you understand why potassium is so important for the resting membrane potential, because it needs to go out. So if you to go out to bring the excitable cells, muscles, and neurons back to the resting membrane potential. Okay, so now there was someone that asked me about page. Professor, the page number provided from the textbook is not matching with the topic. I was looking for calcium ion homeostasis. There were 3 different pages mentioned. Yes, because calcium is so important here. So we're going to be discussing that. Okay, so what I gave you was the pages for this whole set of lectures. Okay, not specifically for today, the whole set of lectures going from lecture 10 to lecture 16. So I already posted all the notes, all the slides, all the material that you need to know for lecture exam 2 is already posted on Brightspace. Okay, any more questions?
I have another question.
Yes, please.
I'm not really sure how to phrase it, but am I correct in assuming that like all of these like cascades of like changes that occur on a cellular level like occur due to emotions? Because we just went over like how muscle contraction like sort of forms, and this would occur like if an animal in the wild like feels fear because it like senses a predator or something. So am I correct in assuming that like you sort
Well,
of feel it?
one thing is emotion, right? That's the fear. That's emotion. That has nothing to do with the muscle. And how is that converted to the animal running away, right? Is that what you want to know?
Yeah, because
So,
I'm like you would only produce like the emotions that or like the chemicals that you need during like fight or flight mode if you felt fear.
right. So what we are talking today is just the motor neuron that sends a signal from the spinal cord to the muscle fiber. Now, that motor neuron receives signals from many different parts of the brain that are going to activate. One is the emotion, which is the amygdala. Amygdala is the part of the brain that is dedicated to emotion. So, when in this case the animal is in fear, it's going to send a signal from the amygdala to the motor neuron. And then from the motor neuron, it will go out to the periphery. So the fear is part of the central nervous system, while now we're talking about the peripheral nervous system going from the spinal cord just to the motor neuron. So we're just focusing on this small area. And now, how will that induce muscle contraction or the movement of muscles? This is what we're going to cover in the next set of lecture, in the next class on Tuesday. Is
Okay.
this clear?
Yes, that does make sense. But then I guess my follow-up question would be like, okay, then like what if this is like just the end of the or like the part of like a middle of the cascade of events, and like what's the beginning?
The beginning? The
Yeah.
beginning is the generation of an action potential, of a signal that can come from different parts of the brain, right? So different parts of the brain are dedicated or receive signals from receptors that are distributed throughout the body, right? So for example, what is the beginning? Let's say you have a flashlight and the light opens all of a sudden. So the eye, which is the light receptor, let's say, will be activated by the photons. And that's what initiates the action potential that then is going to be propagated to many different parts of the brain and different parts, and it's going to result in some cases in muscle contraction, but not in all cases. So the initial is the signal activation of receptors that
Okay.
respond to different stimuli.
Okay, that makes sense.
Okay. Okay, good questions. Okay, guys, so I'll see you on Tuesday. Have a good weekend. Bye-bye.